Um, okay. Hello, everyone. Welcome to another session of Alumni Talk series presented by Biotech Research Club, IIT Madras. Today we have with us Atmika Paul. Um, she's an MS scholar. She got her MS degree from here at the Department of Biotechnology, IIT Madras, in 2018. Um, to give a brief introduction about the speaker, I am personally very excited about today's talk. So Atmika got her BTech degree from LIT Durgapur, and later she got her MS degree from here at IIT Madras, where she worked under Professor Mahalingam um, in cancer biology, in different projects in cancer biology. She got her MS in 2018, and she later worked as a project associate in the same lab for a short duration, and then later she went on to pursue her integrated masters and PhD in International Max Planck Research School for Molecular Biology, University of Göttingen. She's currently pursuing her PhD. She finished her master's program, and I think she's stepping into the PhD program. Um, so her interests are in understanding the molecular mechanisms of cancer biology, which is responsible for cancer progression, and she is also interested in defining therapeutic targets, for the better management of cancer. So thank you so much for accepting our invitation. Over to you, Atmika.
It's an honor. Okay, so hello guys. Okay, if I'm too fast or anything, please let me know. If my internet goes off, well, I am really sorry, but I hope it doesn't. And I'll start. Um, so I hope it's visible. And well, first of all, thank you for inviting me here to talk. It's really an honor to speak as an alumni of IIT Madras. It has been my home, IIT Madras, and my lab, and I loved every second of the 3.5 years I spent there. My lab people were like a family to me, and I would love to answer and help you with anything that you people ever need from me. So I think the poster already told about my topic. The topic is deciphering the role of TGF-β signaling in maintenance of genomic integrity in pancreatic cancer. That's a lot of words, so we'll go to it later on. I was told that I should probably tell a bit about my journey. So I'll tell very briefly about my journey. It's short but a bit eventful. So I did my bachelor's from National Institute of Technology, Durgapur in biotechnology. And I have always been scared that there's too much competition out there and I'll probably never be able to get a good job or, get a good position in a lab. So I did my internships from literally my first year of my bachelor's. So I did a couple of internships, in industrial internships and academic internships in Biotech and at IIT Kharagpur. After that, I was selected as a summer fellow by the Indian Academy of Sciences. And I had to mention this because this is the place from where I was so inspired and motivated to work on cancer in the first place. So there I worked with a very dynamic research group on microRNAs and how microRNAs affect cervical cancer progression. And it was a very good internship program for me. They took us to hospitals. We saw patients suffering there. We saw families suffering there. We saw doctors. We asked their concerns. It was a very interactive program. And from then on, I kind of knew I wanted to work in cancer through my whole life, if possible. And after that, in NIT, I did my thesis on molecular biology, and I wanted to understand how aging happens and the role of reactive oxygen species in aging. So there goes my bachelor's. Now I come to my master's, my good times, so-called. So I was in the lab of molecular virology, At that time, I still like that name better. So LMV, and now it's LMCB, I guess. Yeah, with Dr. Mahalingam, and I had an amazing time with my lab mates. Some of them are there in the lab still now working, and there I worked on several projects, mainly on RASFs, because our lab was focused on RASFs and also GNLs, but I was in the RASF group. And I also worked with my postdoc, Dr. Ananthi, in another project in oral cancer. So I was working on breast cancer and on oral cancer. I presented my work in this beautiful city of British Columbia, Canada, at the Keystone Symposia, and I presented this poster. Yeah, it was my thesis work. And after that, I was working as a project assistant in my lab for 6 months. And then the time of transition started, where I was applying for PhD to different places. And very fortunately, I got selected in the National Institute of Singapore, where I got in with a research scholarship, which is kind of difficult to get in because there are hardly 2 or 3 people selected every year out of 2,500 applicants. And there I was working again on cancer because I am obsessed with cancer. Yeah, so I was working on angiogenesis, and I wanted to see the role of matrix metalloproteinases and how they affect cancer metastasis with this group of Dr. Guruven, but unfortunately it did not work out for me. Well, not everything works out, so it's okay to move on. And after a semester, I left National University of Singapore because and the work environment was not okay for me, and the project was not clinical enough. And very fortunately, I got selected in a university in Europe, in Germany. You would have heard of the Max Planck Institute, and then there's this University of Göttingen, and together they have a program which is the MS PhD Molecular Biology program of the Max Planck Research Society. And I did my MS and PhD coursework there for a year. And so after that, you can either do a master's thesis and then a PhD thesis, or you can do a direct PhD. But I had a very, very long master's thesis over here already in IIT Madras, and I didn't want to do master's once more. So I just went for a direct PhD program. And this is the project that I have been working on since last October. So it's a few months. It's just a few months. Don't expect too much of progress in my work. But yeah, I'll try to explain my work as briefly and as clearly as I possibly can. So, about my project, as you saw from the title of my project, I'm working on pancreatic cancer. Why we need to study pancreatic cancer? It is currently the 7th leading causes of cancer-related deaths across the world. It is the 11th most common cancer in the world. So you might think it's 7th and 11th. Why are we studying this? Because the number of deaths that pancreatic cancer is currently causing, by the year 2040, it will probably be the 2nd leading cause of cancer-related deaths. So we need to study pancreatic cancer. There's no proper treatment available for pancreatic cancer. If you have pancreatic cancer once, the chance that you will survive for 5 more years is less than 9%. So it's extremely aggressive. And once you're detected with the cancer, there's literally very less chance that you will survive at all. So of course, as you can understand, we really need to focus on how to clinically manage pancreatic cancer. Now, I'll give a brief overview of the pancreas once again, just in case we all forget, right? So yeah, so pancreas has an endocrine part and there's an exocrine part. So the endocrine part is the part which secretes hormones, and the exocrine part of the pancreas is the part which secretes the digestive juices. So most of the cancer in the pancreatic cancer happens in the exocrine part, and when that happens, we call it pancreatic ductal adenocarcinoma. I'll shorten it to PDAC. And PDAC accounts for 85 to 95% of all pancreatic cancers. The endocrine cancers are kind of less common. So we are focusing more on the exocrine form of pancreatic cancers in our project. So now, why is it so challenging to treat pancreatic cancer? Because the warning signs or the symptoms of pancreatic cancer are very generic. So you see symptoms like you have in jaundice, like yellowing of the eyes or the skin, you have back pain, and you have diabetes. And very often the doctors don't associate it with cancer. So by the time it's detected, this cancer, it's already very late. So it's most of the times it's detected in stage 3 and stage 4, and then it's very difficult to treat this cancer altogether. So as I already told before, it's a very aggressive cancer. It spreads very easily to the vital organs of the body. The cancer is highly heterogeneous, so it's very difficult to treat. So what I mean by heterogeneous is that the cancer cells differ within the same patient, and they differ from patient to patient. So a single form of treatment does not work for the same patient or for different patients. So it's very difficult to clinically manage this cancer. And when you actually give a therapy, the cancer shows a very high resistance. So there's an extremely high therapeutic resistance. So we wanted to know why pancreatic cancer is so challenging. Why does it pose all these challenges that we discussed so far? So we thought, what if chromosomal instability is the answer? So now I'll explain chromosomal instability a bit. So this is the famous hallmarks of cancer diagram. I am not sure how many of you are familiar with it, but I guess LMV people are pretty familiar with it. But yeah, so hallmarks of cancer are a set of characteristics which the cancer cells gain over time, which help the cancer cells to maintain their cancer-like properties. So one of the major hallmarks of cancer is genome instability. So what is genome instability? If you break the word, it's instability in the genome, where there are frequent mutations and rearrangements that keep happening in the genome. So there are three main types of genomic instability: microsatellite instability, nucleotide instability, and chromosomal instability. And as you can see from this diagram, chromosomal instability accounts for almost 75% of all the genomic instability, and that's what we'll talk about today. So there are two types of chromosomal instability. It can be numerical chromosomal instability, or it could be structural chromosomal instability. So now to define chromosomal instability, as I already told, it is a form of genomic instability where there are frequent changes in the cell's chromosomes. So it could be seen as a gain or loss of whole or a part of chromosome, which is called aneuploidy, as you probably know, or you could see complete chromosomal rearrangements like translocations, etc. So when this happens, as you can expect, over time, the cancer cells become more prone to become more cancerous because they don't respond to the usual treatments, and the cells keep accumulating these genetic changes over and over time. And what happens is these cancer cells which have these genetic changes are able to survive the conditions better, the carcinogenic conditions better, and therefore cancer progresses even more. But it's still not understood whether chromosomal instability is a cause or consequence of cancer, but what we believe that it could be both. So there are various reported mechanisms for chromosomal instability, and I will not go into much details, but then I guess there are some points which would be easy to understand. For example, if you remember your cell biology classes, the normal cells are generally bipolar, like this. But then if there's a multipolar spindle, then of course the chromosomes will be pulled in all directions at different dynamics, and therefore it would lead to chromosomal instability because the chromosomes won't know which daughter cell to go to. So similarly, there are various other mechanisms, like if you have a defective spindle assembly checkpoint, or if our microtubules start moving more frequently, or if they start moving less fast than they generally do. That is, if there's an aberrant mitotic spindle dynamics, then it would lead to chromosomal instability. So I would like to explain this a bit more. So when our cell divides, I guess you would remember the spindle apparatus. The spindle apparatus functions with the help of microtubules. Now, if these dynamics go bad, that is, if they go aberrant, then the microtubules start moving faster. If they move faster, the chromosomes can't divide properly in the right direction, and therefore they might go to a different daughter cell than they're actually supposed to, and this would lead to a chromosomal rearrangement, which would lead to genomic instability. And this picture would kind of help to explain that a little bit. So for example, if the microtubules start moving faster, then a bit of the chromosome is left behind, and this cell has already divided, and this is already left behind, and this is probably supposed to be in this daughter cell, in the other daughter cell, and instead it is coming to the second daughter cell, and therefore this daughter cell is left with an extra chromosomal strand, and this would lead to genomic instability. So now, chromosomal instability and why we are studying it in cancer. So almost 60 to 80% of all human cancers show chromosomal instability. It is reported in a variety of cancers, as you can see by the various ribbons, representative of the various cancers. Why we are studying it in pancreatic cancer? Because 97% of pancreatic cancers have been shown to exhibit chromosomal instability. So we really need to understand what are the mechanisms that lead to chromosomal instability in pancreatic cancer. So with that, we come to our research question. We know what are the intrinsic mechanisms in the cell that lead to chromosomal instability. We know chromosomal instability is a major phenomenon in pancreatic cancer. We know that this phenomenon leads to the heterogeneity, metastasis, therapeutic resistance, poor patient outcome. But what we don't know is what are the extrinsic signaling mechanisms that lead to chromosomal instability in pancreatic cancer. So keeping that in mind, we did some literature search and we wanted to check what are the major cellular mutations or what is the molecular landscape in pancreatic cancer, so that we have an idea of what are the signaling mechanisms that might be critical to maintenance of genomic integrity in pancreatic cancer. And what we found is that there are almost 12 signaling pathways which are found to be aberrant in pancreatic cancer. There are 4 genes which are found to be majorly mutated in pancreatic cancer. And one of the major pathways that is mutated in pancreatic cancer is the TGF-β signaling pathway. One of the major genes that is mutated in pancreatic cancer is SMAD4. So SMAD4 is a key player of the TGF-β signaling pathway, which I'll show you later on. I'll show you the pathway later on. And SMAD4 is found to be lost in almost 60% of pancreatic cancer samples. And there are several other genes in this TGF-β signaling pathway, and pancreatic cancer patients show a range of mutations in all these genes of the TGF-β signaling pathway. So this kind of justifies the fact why it is important to understand if aberrant TGF-β signaling has a role to play in pancreatic cancer progression. So this is the pathway. So I'll explain the pathway very briefly. It's a bit complicated, but yeah, I'll simplify it for you. So the TGF-β signaling pathway has a canonical pathway, which is the more common pathway, and there are several non-canonical pathways, which are the less common ones. So what happens is, as usual in any pathway, so the ligand sits on the receptor. So the TGF-β ligand sits on this TGF-β receptor. It leads to phosphorylation of the receptors, the TGF-β receptor 1 and the TGF-β receptor 2. This phosphorylation leads to phosphorylation of SMADs, which are also called the receptor SMADs. So SMAD2 and SMAD3 are phosphorylated. And once SMAD2 and SMAD3 are phosphorylated, they form a complex with SMAD4. And only when it forms a complex with SMAD4 can it go into the nucleus. It can translocate into the nucleus and help in gene transcription of its target genes. So from this, you can already appreciate that if SMAD4 is not there, this entire gene transcription that is supposed to happen from the TGF-β signaling pathway is lost. There's no transcription happening anymore if SMAD4 is not there. Yeah. Now let's come to the non-canonical pathway. So apart from this SMAD dependent pathway that we saw, TGF-β can sit on its receptor and it can activate several other common oncogenic pathways also, like the RAS-RAF pathway, the PI3K-AKT pathway, and all the classical oncogenic pathways that we study in our bachelor's and master's. So what is the effect of TGF-β signaling? So if you have attended developmental biology classes, you would know it is one of the major 4 developmental pathways. It is involved in a variety of cellular processes like cell cycle arrest, apoptosis, inflammation, angiogenesis, proliferation, invasion, etc. And trust me, 1,000 other cellular processes. But then if you notice here carefully, you will see that there are some activities which are tumor suppressor type of activities, and there are some which are kind of oncogenic. So TGF signaling can a tumor suppressor role, and it can also have an oncogenic role. depending on the stage of the cancer. So when the cancer, when it's a normal cell or when the cancer is at a very early stage, it tries to suppress the cancer, and then when the cancer progresses, it actually tries to help in cancer progression, because over time we see loss of SMAD4 and several other mutations which kind of start favoring the non-canonical pathway of the TGF-beta signaling. So I guess that's a very brief overview of the pathway. Now, to make a connection of the whole project, so there is some literature already available which tells that TGF-beta can play its role as a critical regulator of genome maintenance. So why we are talking about genome maintenance? So if genome maintenance is lost, it will lead to genomic instability, right? So if the TGF beta signaling is aberrant, then the genome maintenance is lost and it would lead to genomic instability or chromosomal instability. So there are several papers which have reported that TGF beta can play a role in stopping mitotic abnormalities. It can play a role in the DNA damage response pathway and in regulation of microRNAs, and therefore it can regulate the genome. So we already know that TGF beta has some role in genome maintenance. So with all this information in mind, we formulated a hypothesis where our question was, what are the extrinsic signaling mechanisms which regulate genomic instability or chromosomal instability in pancreatic cancer? And currently our question is TGF beta signaling one of the critical extrinsic pathways which lead to chromosomal instability in pancreatic cancer? So with that, we formulated some research questions. Our first question was, is TGF beta signaling actually altered in pancreatic ductal adenocarcinoma or PDAC? Our next question was, do pancreatic cells actually show chromosomal instability at all? And our third major question was, does TGF beta signaling play a role in microtubule dynamics and chromosome missegregation in PDAC? So I don't remember if you noticed before, but I already told that abnormal microtubule dynamics and chromosome missegregation are signs of chromosomal instability, and these are the classical ways you kind of predict that a cancer cell is chromosomally unstable. So we want to see if TGF beta signaling has a role in these two phenomena. So this is our research methodology where we do cell culture work, of course. So we have some control cells like the colorectal cancer cell lines and the retinal pigmental cell lines, and we have a panel of pancreatic cancer cell lines, which are these three. And then we do molecular and cellular analysis via Western blotting, immunofluorescence flow cytometry, and then we do functional analysis. For us, functional analysis involves checking the microtubule dynamics, checking whether we see missegregation or lagging chromosomes. So if you remember, I showed you a picture before where there was one chromosome bit that was lagging between the two dividing sets of chromosomes in anaphase. So those are lagging chromosomes. And we also want to do karyotyping. So karyotyping can also tell you whether a cell is aneuploid or whether it's euploid, right? So these are our functional analysis. So I'll go very kind of fast over the results because yeah, it can get pretty boring, trust me. So first, is TGF beta signaling altered in pancreatic cancer? So we did a few Western blots. I will try to briefly run you through the Western blots. So this RP1 are retinal pigmental cells, which are our control cells, and these three are our pancreatic cancer cell lines. So how do you detect whether TGF beta signaling is high or low? So more the levels of phosphosmad2, more is the signaling. So as you can see, compared to the normal cells, the phosphosmad2 levels are lower in all the pancreatic cancer cell lines, which already tells us that PDAC cell lines show a lesser or attenuated endogenous TGF beta signaling as compared to these normal cells. So we already know that pancreatic cancer cells have altered TGF beta signaling status. Next, these cell lines, BXPC3, these show SMAD4 loss, as you can see. So as I told you before, again to remind you, 60% of pancreatic cancer patients show SMAD4 loss. So we felt that this cell line could act as a model for those, as a model cell line for those patients. who show SMAD fold loss. And also among the other mutations that we see in pancreatic cancer, major finding is that many of them show reduced expression of the receptor itself. And if you see here, these cells MIAPACA2, they show reduced levels of the receptor as compared to the other cell lines. So this cell line can act as a model cell line for the patients who show attenuated levels of the receptor. And these are our findings. And then next, so we wanted to determine what is the optimal concentration of TGF-β treatment that we should give on the cell lines, so that we always select the maximum concentration, so that we always select the maximum amount of response. So these are the 3 cell lines that we talked about, the PDAC cell lines. And if you can see, the highlighted part shows the maximum level of SMAD2, phosphosMAD2, which shows that this particular concentration is the one which we should use for our experiments, because we want to see what is the maximum amount of response, of signaling response that these cells have when we are treating them with an external ligand. So this is just a factual Western blot. So we can see that different cell lines require different amounts of ligand and different durations of treatment. And we should be careful as to how long we should treat them with the ligand. Next, our question was whether the PDAC cells or the pancreatic cancer cells actually show chromosomal instability. So first, we wanted to check mitotic microtubule dynamics. So what we do here, I'll try to explain you briefly. So we have our pancreatic cancer cells. We transfect them with EB3 GFP. So EB3 is end binding protein, which binds to the tips of the microtubule. So it binds to the growing tips of microtubules. And when it's tagged with GFP, you can see the microtubules moving, and you can see the growing tips of the microtubules. So of course, you will see a bipolar spindle in the beginning, and then we add an inhibitor called DME. So DME is a kinesine inhibitor. It is a mitotic kinesine inhibitor. So when you add this mitotic kinesine, these spindles are not, the centrosomes are not able to go to the opposite poles, and then they become a monopolar spindle. So it's easy for us to calculate the dynamics of the growing microtubules. So we do live cell imaging of these monopolar spindles, and thereafter we calculate the dynamics of the growing microtubules. And do stop me whenever you feel like, if it's a bit hazy to you. So with that, so this is essentially how these videos look like. I guess you can see these are live cell videos, and you can see the growing tips of the microtubules of a mitotic cell. So for us, HCT116, which is a colorectal cancer cell line, is a control cell line because it is known to be a chromosomally stable cell line. This was already done in my lab. We already know that these cells are chromosomally stable and they show lower mitotic microtubule dynamics. And there was another control cell line, which is also a colorectal cancer cell line, already shown from our lab, that these cells have higher mitotic microtubule dynamics. And when we wanted to check the dynamics for our pancreatic cancer cell lines, all the pancreatic cancer cell lines have increased mitotic microtubule dynamics, which matches that of SW620, which is a chromosomally unstable cell line. So I guess it's fairly safe to say that all the pancreatic cancer cell lines have increased mitotic microtubule dynamics, and hence they might actually show chromosomal instability. So for our experiments, we use Taxol as a control. So Taxol, as you know, is a compound which stabilizes the microtubules, and hence when you add Taxol, it is supposed to reduce the dynamics again. And when we treat these cells with Taxol, we can see that there's a reduction or rescue of the increased mitotic microtubule dynamics. So from this, we concluded that our pancreatic cancer cells indeed have increased microtubule dynamics, which is more comparable to the chromosomally unstable cells. And it might be fairly safe to say that these are chromosomally unstable. Next, there are several other mechanisms of chromosomal instability, and one of them, as I told before, was centrosome amplification. I mentioned before that if you have multipolar spindles, then you would have abnormal dynamics, and hence it would lead to genomic instability, and multipolar spindles result due to centrosome amplification. So you have more centrosomes, hence you have the multipolar spindles, right? So what we saw in our pancreatic cancer cell lines, so our P1 is our control, and we have our pancreatic cancer cell lines, and all the pancreatic cancer cell lines show increased centrosome amplification as compared to the normal cells. Especially the cell line PANC1 has very high levels of centrosome amplification. I'm sorry for the noise. I'm in my home, so yeah, I'm sorry. So this is how a multipolar spindle kind of looks like. And if you see more than two centrosomes, then it's an example of centrosome amplification. So as you can see, instead of a bipolar spindle, we have a multipolar spindle here. So I think it's fairly safe to say that PDAC cells show significantly higher levels of centrosome amplification, and this might be one of the reasons why pancreatic cancer cells are genomically unstable, because maybe pancreatic cancer cells have higher levels of centrosome amplification than normal cells. Next, we wanted to see the number of lagging chromosomes. So what is lagging chromosome? I'll remind you once again. So if there's a chromosome which is lagging behind, when all other chromosomes have already segregated to the two poles during anaphase, it's a lagging chromosome, because it doesn't know where to go, and it goes in one of the two poles, and it leads to improper segregation. So lagging chromosome is essentially a sign of chromosome missegregation. And HCT116 is again our control, which is a chromosomally stable cell, and you hardly see 2 to 3% of chromosome missegregation in HCT116 cells. But in the other cell lines, which are our pancreatic cancer cell lines like MIAPACA2 and BXPC3, you see very high levels of chromosome missegregation as compared to the normal cells. SW620 was also our control cell, already shown from our lab that these are also unstable cells which show increased chromosome missegregation, almost 8 to 10%. What is interesting to see here is that pancreatic cancer cells show very high rates of missegregation as compared to the even colorectal cancer unstable cells. So it's safe to conclude that pancreatic cancer cells show a very high percentage of lagging chromosomes or chromosome missegregation. I would also like to tell how we detect it. So we do immunofluorescence, where we stain with DAPI, and we stain with kinetochore proteins or centromeric proteins. And if there is some centromeric or kinetochore protein which is lagging behind, you just count the number of cells which show this type of phenomenon, and you calculate the percentage. So yeah, pancreatic cancer cells show a very high percentage of lagging chromosomes or chromosome missegregation. So with this, we come to our third question for now, which is whether TGF-β signaling has a role to play in mitotic microtubule dynamics and chromosome missegregation in pancreatic cancer. So we have some work that's done. We don't have a lot of information on this yet, but yeah. So let's come to our first cell line, MIAPACA2, which is a pancreatic cancer cell line, as you have seen before. So when we inhibit the TGF-β signaling pathway using TGF-β receptor inhibitor, we see that the mitotic microtubule dynamics go down as compared to untreated cells, which already tells us that TGF-β signaling is critical to maintenance of proper microtubule dynamics in these cell lines. Next, we wanted to check if a similar phenomenon is seen in chromosome missegregation. So we did a similar form of treatment where we treated the cells with the inhibitor in these two PDAC cells, MIAPACA2 and BXPC3, and compared to untreated, we see reduced chromosome missegregation when we treat with the inhibitor in both of these cell lines, which tells us that inhibition of the receptor or inhibition of TGF-β signaling leads to reduction of mitotic microtubule dynamics in MIAPACA2 cells to almost the normal levels. So this kind of suggests that maybe inhibition of this pathway could be a way to reduce the abnormal genome dynamics that we see in pancreatic cancer, and that might be a way to the cure, and it might reduce the therapeutic resistance that we actually see in pancreatic cancer. And further, we wanted to see what is if it's the canonical pathway or the non-canonical pathway that is important for this maintenance of microtubule dynamics. So if you remember, the canonical pathway of TGF-beta signaling was a SMAD dependent pathway, and I told you before that SMAD4 is a critical player in the canonical pathway. So when you knock out or knock down SMAD4, or if there's a SMAD4 loss, then it would try to shift to the non-canonical pathway, because the canonical pathway can't go on at all without SMAD4. So what we see is when we knock down SMAD4 with siRNA, there's an increase in microtubule polymerization rate, which suggests the fact that maybe when there's no canonical pathway happening, the pathway shifts towards the classical non-canonical pathways like RAS, RAF, and PI3K-AKT pathway, all of which are known to contribute to chromosomal instability. And that might be the reason why SMAD4 loss pancreatic cancer patients see so much of aggression in their biology, in their cancer biology. So we have a lot of research questions that we still need to address, of course. So we first want to see whether inhibition of or activation of the TGF-beta signaling pathway has a role in the long term on genome dynamics in PDAC subtypes. So what are classical and basal PDAC subtypes? So these are the classification which is currently being used at a clinical level to treat pancreatic cancer patients. So we wanted to see whether this classification is also reflected in terms of genome dynamics studies. Next, we want to see what are the major non-canonical pathways that contribute to genome dynamics, altered genome dynamics in SMAD4 deficient PDAC cells. Because if SMAD4 is not there in PDAC, there's no canonical pathway. Then why is there an altered genome dynamics in these cells? So we want to see what are the major non-canonical pathways there. And lastly, we want to see if this aberrant TGF-beta signaling dependent change in genome stability can actually be translated at a clinical level, and whether we can use this as a strategy for patient treatment and for deciding what kind of treatment to give to the patients. So we want to see whether PDAC patients actually have altered genome dynamics, and we want to see if the patients who have altered genome dynamics show an aberrant TGF-beta signaling pathway. If we already know that, maybe there's a way to treat them already, or there's a way to diagnose the cancer at a very early level. And this is an interest of my lab, which my professor is interested in, whether SMAD4 loss triggers replication stress. But yeah, we'll look at it later on. Let's say. And with that, I finish off. I hope I didn't bore you guys too much. And I want to acknowledge everyone who has helped me out. So my thesis advisor, Dr. Holger Bastians, my committee members, and I am part of this consortium, Pancreatic Cancer Consortium, and they give us all the facilities, and I'm also a student representative of this consortium, and my Max Planck Society for giving me the fellowship. Of course, I can't survive without money, and Max Planck Society, and my colleagues from my lab, and because my professor's wife's lab is also in the same floor, so colleagues from there, and my ITM lab mates, and Dr. Mahalingam, who has supported me a lot. I can't even begin to talk about it. And my National University of Singapore lab mates, because they taught me a lot about life and how to work in lab, and how to stay abroad, and yeah, family and friends. And that's all. Currently, I do a lot of stuff apart from lab work. I travel a lot. I do photography, I dance, I paint, and of course, I work in cancer, and I always try to spread some awareness about cancers. And that's all. Thank you so much for your time.